So yeah, so the Jupyter notebook that we're running for me and Daniel's sessions are this Evol and SignDy one. But we're going to be starting with the actually the second section, the SignDy section.
That's the same notebook that we can get from the Prodi website, right?
Yeah.
Okay, thanks.
Yeah, from the website or from the Google Doc. Actually, I think the Google Doc just links to the Prodi website. Yeah.
So hello. Okay. Can you just, if it is not gonna take more than 30 seconds, can you just please, yeah, like let me know how to run it, because I could not run it, you know. So just download it and then.
Yeah. So just do save link as should. Yeah. So if you just right click that and do save as, it will should save it as the IPy notebook. Or they should be on the website, right, Burak?
Yes, exactly. Just go down, click here in the workshop.
Oh, latest workshop.
Yeah, I could. Yeah, I have it. And then how can I use it?
Do you have your Python environment set up?
Right. Yes, I have it.
So do, I guess, type Jupyter space notebook space and then the name of the notebook. So in this case, it would be Jupyter notebook and then Prodi underscore, etc.
Okay.
Did you get it working?
No worries, you can start. Okay.
Oh, okay. I'll go ahead and start. If one of the other Bahar lab people want to try to help out, maybe it's in a breakout room or something, that'd be great. Okay, so we're going to start off by just doing our normal imports, so Prodi and PyLab, and then like in my section yesterday, we want to set this path to PDB folder because we're going to be downloading PDB structures again, just so we don't want to get everything all messy. And then, so we're going to just scroll down to section 2, so doing signature dynamics. So in this section, we're going to be doing GNM analysis of entire protein family, which I believe Pemra talked about the SignDy section today in the lectures. So we're also going to just do one more import here. That's just a built-in one, so it should work for everyone. And so what we're going to start off by doing is doing a Dolly search. So what this is, Dolly is a server that has a number of protein families. So basically what we're going to be doing is using this built-in function of Prodi to query the server with this PDB code and then the chain that we're specifically interested in. So if we just run that, it has been running pretty quickly recently. Okay, yeah. I think it might be because it caches results. So since I've been running it a number of times testing, it should be in there. But if everyone wants to just run that quickly and see if you all get your results pretty fast. And if anyone's not getting results, could you go ahead and let us know too before we move on?
Sorry to interrupt you. Is there anyone who wants to go to breakout room? I see 6 breakout rooms. Are you all see that? Let me open them all. So I guess you should be able to join them if you want. Do you see all the breakout rooms?
I don't know
Yes,
if everyone can just join. We might have to assign them if you have
I
questions.
made it accessible for everyone, but if you want me, just let me know. I can assign you to any group you want. Just 1 second.
Okay. So I haven't seen any messages about people having problems so far. So like the notebook says, sometimes this Dolly search can take a while if their queue is backed up. So if it times out, there's just this other function that you can do is to just wait periodically and then keep querying it until you finish it. You can run that. It should just run automatically since at least for me, I already have results. And then, okay, these 2 lines you don't have. So next what we're going to be doing is filtering our results, basically to get rid of sequences that are either too similar or structures that are too similar. And so if we run this filter, we get rid of almost all of our results, and we have 166 structures. And what we're going to be doing next is run this get mappings function. And so what this does is it's going to map all of the sequences onto each other. And we're going to use that for when we do our build PDB ensemble method. So you just run that, and then what we're going to be doing is the parse PDB method and give it all the PDBs that remain after being filtered. So that's going to download all those structures from the PDB. Take a minute. And then once this finishes running, we're going to use the same function we did yesterday for the ensemble. So build PDB ensemble. We're going to give it our list of structures. And then we're going to tell it specifically to use this mappings that we got from the Dolly function. And then we're just going to give it the PDBs for the labels. So then it's going to attempt to map all of these structures to the reference structure, which is the one that we use for querying the Dolly server. I don't know why this is just printing out everything and using up all this space. Some structures it's not going to be able to map, so it will just ignore those, but you should be left with 72 structures. And then we're going to just save this ensemble so we can load it later. So then if you want to come back to using this, you don't have to keep querying it and aligning and everything. Okay. So is anyone's code still running up to this point? If you could just like raise your hand or something in the in Zoom. It looks like we're almost done. Okay, I don't see any more hands. Okay, so I'll keep going on then. So basically now what we're going to do is this line just loads that ensemble back up. And now we're going to use this calc ensemble ENMs method to perform, we just want to do GNM on this ensemble. And what this trim keyword does, telling it to reduce, is for any sections of the structures that don't map onto each other, we're just going to get rid of them. So we can run that, and it's going to just calculate our by default 20 modes for each structure in our ensemble. And then what it's going to do then is it's going to, it says matched. So what that means, it's going to go through each mode and put them so they are all in the correct order. So sometimes when you're doing your ENM analysis of different conformations of the same structure, the modes might be not correlate perfectly. But this is going to go through and figure out which modes from all of the structures correlate to each other and make sure they're all in the same order. And then we can just save this ensemble as well, and then reload that so we don't have to keep running it. So I think Burak yesterday was talking about how like all of the Prodi data structures are, that's because you can just slice them. So if we wanted to get the first mode set of our GNMs, so we do GNMs bracket 0. And so what this does, you can see it returns a mode set. So we just by default calculated 20 modes from this PDB structure, the 3H5V chain A. We can also select a specific mode from this mode set. And so this is getting all of the, sorry, this is the first mode from all of the mode sets. And then just like you would expect, you can slice any combination of those or just get a specific mode from a specific mode set. And so a mode set is just the GNM results from one specific conformation of the protein. So now what you're probably going to want to do next is look at the actual signature dynamics, as we saw in the lecture this morning. So we can just use this show signature mode. And what we're doing here is just looking at the first mode of all of the structures. So we gave it. So you can see that this is the dark blue line is just the average value at that residue, that specific residue. The dark blue is the standard deviation, and the light blue shaded area is just the total, the highest and lowest value at that position. So we can see where there is a lot of conserved regions where there's not much variance in this structure, in this protein family. And in some of these regions, there's a lot more variability in the dynamics of this first mode. So we can also, for example, look at the mean squared fluctuations of these first 5 modes using this show signature square flux, and then giving it the first 5 modes of all the structures, and also look at the cross correlations of this entire mode set. So we can see as we would expect, the correlations are highest along the diagonal, but then we also see some off diagonal areas where there are more highly correlated motions in the first 20 modes for parts of the protein that are more distant sequentially. And
Quick question. Or am I crazy, or the file that you're working on is not the same that the one that we downloaded from Prodi? Like, I cannot see any of the things you're doing with the file I downloaded from.
Me too. That's why.
are you running them at the same time?
No, I don't see the cells that you're working on in the file that I downloaded. It has different stuff, each cell.
Yes.
I think it is the same notebook, but it's like the second part of the notebook. So if you scroll
Yeah.
down on your
We
notebook,
are,
there is a
sorry,
part
we-
that says signature dynamics analysis with SignDy. And
yeah,
so
we're
that has
in
all
this
the correct
second
ones.
section.
Yeah.
Oh, I am crazy then. Thank you.
Yeah, sorry, these two are combined, but we swapped the order this time because Daniel is also doing the next section. So we're starting with the SignDy analysis.
Okay.
Thank you.
Thank you.
So what we can also do is look at specific members of the ensemble. So what this function, this graph is doing is showing the variance in different modes. And so it's that we can look at the eigenvalues and eigenvectors of the ensemble, the mode ensemble, just doing these get eigenvals, get eigenvex functions, which as you can see just spit out these big arrays, but give 7220 is the shape as we would expect. So it's over 72 modes and our 20 or 72 structures in our 20 modes. And then for the eigenvectors, the 376 is the number of atoms in the shape of this array. So a important value that we can look at with these ensemble is the spectral overlap. And so this is the overlap in the dynamical overlap. So the similarity in the dynamics of any pair of structures. So what this plot is doing is, so we generated our spectral overlap matrix using this calc ensemble spectral overlaps function, and we gave it, so GNMs, we gave it every structure and just the first mode. So we do show matrix. So this is looking at spectral overlap. So from 0.5, I guess, to 1. So a higher overlap is a higher level of similarity in the dynamics of the first mode. In each row and column is a different member of this protein family. So similarly, we can we can also make this a distance measure, where so a lower value is more similar, which is what we see here. But these are very basically the same plot. So we can use these metrics in order to get a tree. So what this tree will do is calculate how similar different members of the family are based on this metric. So based on this dynamical similarity metric. And for some reason, it is making it very tiny. When everyone else runs it, does it show up as very tiny also?
Yes, for me.
Okay. Am I? Okay.
Kind of related to that. Can I ask a quick question?
Yeah.
A way to save these charts and graphs as like a .ai file, so that they can be cleaned up or polished up, or is it just .png files that you can save as?
Is that an Illustrator file?
Yeah, Illustrator, Photoshop, any sort of vector. I guess I would prefer a vector file.
So if you're saving them, it generally depends on what matplotlib, which I believe is the plotting function that we use for everything. I think matplotlib will allow you to save as like an SVG. Or
gotcha. Okay.
But so yeah, so once if you like generate this figure, for example, you would do if you have plt.save fig, and then you do .svg or whatever, and then for example, something like that. And I'm pretty sure matplotlib allows you to save as a vector file, but you might have to go just search their search like from them specifically, because I don't know if any of the built-in methods in Prody will do that.
May I have a question?
Yeah.
Let's suppose I have an ensemble for my molecular dynamic simulation. I can use this type of dynamic analysis to analyze my ensemble and separate and cluster the different conformation.
Yes, you should be able to, if you give them, separate them into like different PDB files, you would be able to load those all separately into an ensemble, and then perform like GNM analysis on the entire ensemble.
Okay, thank you.
Okay. Sorry, I'm just looking through the chat, trying to catch it. Okay. So I think might be missing something for this show tree. Usually it's
You can put the figure and fig size that it's on the cell below to increase the size of the show tree. It worked for me. I just copy pasted that.
Okay.
And it worked.
Okay.
Actually, Matplotlib, you can change whatever you want. You can change the ticks, the size of the fonts, and whatever.
Okay. Yeah, that did work. I don't know why that wasn't in there. Okay. Yes. So this will show our tree that we generated based on the spectral dynamics, the overlap similarity. And then what we can do is use this reorder matrix function. And what this will do is try to cluster them together based on our overlap matrix that we generated previously and this tree that we just generated. And so this will just give us new indices, and we can give that to our plotting function. And now we see some nicer structure, some more conserved blocks, as opposed to what we were looking at previously, where everything was just in whatever order the Dolly ensemble was downloaded in. What is this one? Oh, I don't know. Okay, sorry. I don't know why this is necessary. Just flipping it. Okay, so this one, what this plot is doing is we give the show matrix function our tree, and so what this is doing is it along our y-axis, it's actually plotting the tree that we generated next to our heat map of our spectral overlaps. And then these two cells are just reordering our ensemble and our GNM ensemble based on our this reordering that we did, and then getting just a new list of the labels. So similarly to what we just did, looking at our spectral overlap, we can make plots and compare our structural ensemble based on either sequence similarity or structural similarity. So to start off, we're going to be looking at our sequence similarity. So one of the functions that this ensemble has is get MSA, which will give us a multiple sequence alignment. And so we give that multiple sequence alignment to this build SignDy matrix function, and then just basically take the inverse of that, do 1 minus our SignDy matrix. And then if we give that to the show matrix function, it will plot our sequence similarity in the same order now as we're looking at from our reordered matrix. So basically what we're seeing is some blocks of sequence distance. So a low level for this Hamming distance means these blocks are more sequentially similar, whereas these red values are less similar sequentially. So we wanted to reorder this ensemble similarly to what we did before. We can just do the same sort of thing, calculate tree, and then reorder our ensemble and plot that again. So slightly cleaned up compared to before, but we can still see these big blocks of very similar sequences, as well as a few sequences that are have a medium level of similarity, and these two clusters which are not similar to basically any other member of the family outside of their own cluster. And then lastly, we're basically going to be repeating this analysis based on the structural similarity. So what we're going to be doing is calculating the RMSD of each pair of our ensemble. So just getting the how similar each pair of structures is, and then plotting that. So again, low value for RMSD is more structurally similar, high value is less structurally similar, though this is topping out at around 4 angstroms, which for an entire protein is still pretty similar. So I guess this is only the alpha carbons. So again, we can get our RMSD tree, and we can reorder that if we want. We can also look at all these trees. So we did our tree based on RMSD. We can look at all of these clusterings side by side here. So look at the trees generated by sequence similarity, structural similarity, and the dynamical similarity from our spectral overlap. Though if you wanted to actually compare them better, again, yeah, you should make it larger so you can actually see the PDB IDs. And then we can also reorder all of our ensembles based on the same ordering and look at the similarity of them next to each other. So everything here is ordered based on sequence similarity. So we can see that these areas of sequence similarity don't necessarily lead to like highly structured structural or dynamical similarity plots. And then as a last note, it says here that the analysis is quite sensitive to how many modes are used. So as you, in specifically the dynamics, so as you increase the number of modes that you use for this analysis, eventually you can use the entire mode set generated if you wanted, but that will start approaching the same plot of the structure value. And so it's basically just resetting this PDB folder path because we don't want to keep downloading stuff there. And so that is it for the SignDy analysis section. Are there any further questions?
Yes, I have a question. So all of our dynamics analysis was done from the modes using GNM, right?
Mm-hmm.
So is there a reason why we use GNM instead of ANM?
So in this case, one of the reasons is that it's just faster. So if you're calculating modes for an entire ensemble of proteins, especially if you're trying to do the entire set, just doing GNM is faster than ANM. Additionally, I believe GNM actually has better agreement with experimental evidence. However, there are some specific things that analysis with ANM gives you, such as actual directionality, or not directionality, gives you actual ability to visualize the modes of motion that GNM doesn't. But if you're only interested in comparing the actual, sorry, just going to scroll to the, like looking at just the profiles of the modes, then all you need is the GNMs.
Okay, thanks. Also, can I ask you something about the plot underneath the cross correlation?
Yeah.
So it has, that's the number of residues, that's 3. Oh, okay.
It's like 376.
Okay, but that's for the whole ensemble. So my question goes to the point that if you are comparing different structures, they don't necessarily have the same number of residues, do they?
So, okay, so when we built our mode ensemble, so this
Yeah.
calc ensemble ENMs, what it's doing with this, so this trim argument, we're telling it to reduce any residue that doesn't map to the reference, so or the shortest one, it's going to just get rid of that. So all of the every single mode set in this is going to have the same number of atoms.
Oh, okay, yeah.
So even if they're different structures.
Okay, yeah, that makes a lot of sense now. Oh, thank you.
Yeah. Okay. Is there anything else? I don't see any other questions in chat. So if not, I'll give it over to Daniel, and he will take over for the Evol section. Daniel, are you there?
Yeah, I'm here.
Okay. I'll stop sharing and you can take over.
Okay, great. Thanks, Bentley. All right, let me get set up here. Okay. All right. Okay, can everyone see my screen?
Yes.
Okay, thank you. All right, everyone. So I'm going to go over the Evol section, which is actually in the Jupyter Notebook is actually the first part. It's actually the first section, but we went in reverse order because I'm going to be giving the tutorial on the membrane A&M after this. And you'll notice that my Jupyter Notebook looks a little different. I have it in this dark mode, and I unfortunately forgot how to convert it back, but it's the same Jupyter Notebook, and I will back and forth between my notebook and the one that's available to you all on the workshop website, just to go over some of the figures, because they're going to be kind of difficult to see from my notebook. All right. So to get started, we're going to go over the Evol API that Prody offers. Okay. And this is a sub package that allows you to fetch and parse, also refine multiple sequence alignments, and you can also use it to calculate certain residue level quantities, such as estimates for conservation and coevolution. All right. And you could even calculate global or sequence level quantities such as percentage sequence identity. So I run my first cell. All right. So to get started, you need to make sure that you have this PDBs folder in your working directory. Okay. So we can we can configure Prody to put all the PDB files in a particular folder in our working directory. And so that's what I'm going to do here. And now to get started, so there are several publicly available resources out there that provide multiple sequence alignments for different protein families. And one of those is PFAM. And Evol has several functions that helps retrieving this sort of data in a very convenient way in Python, all right? And so more about PFAM. PFAM is protein families database that provides the multiple domains, okay? And this is, PFAM is convenient to use because it's sort of connected to other databases out there like UniProt and the PDB, all right? Now, when you're going to look for your PFAM or a specific protein family in PFAM, you're going to need the PFAM accession ID, okay? So whenever you're going to look up the multiple sequence alignment for a protein of interest, you're gonna have to figure out its PFAM ID. We provide that for you here, all right? So once you have that using fetch PFAM MSA, it's really easy. All right. So this is saying that, you know, I was able to retrieve the multiple sequence alignment that's in a dot, that has this dot STH extension. So there's different file formats for multiple sequence alignments. The one that I come across most often is the Stockholm format, but there are others out there. There's FASTA, Clustal, and to basically access the data in these files, we're going to use parse MSA from Evol, and this function can handle these various file extension types for multiple sequence alignments. All right, and now we have our data in this MSA object of ours, and we have nearly 1,500 sequences, and there are nearly 348 residues or columns in this multiple sequence alignment. Now this object is sliceable, so you can index the alignment to extract the sequences which will correspond to the rows. Okay. You can also slice the residue positions or the columns of the multiple sequence alignments. All right. And I'll give you back another object, and it's letting us know it's letting us know what we selected. You can also select out individual sequences from the multiple sequence alignment. All right. So by using index 0, we're taking out the first sequence here, okay, and lets us know like the protein name. You can look this up use this to look up the protein on UniProt, right? And we have its length, residues, and the number of gaps. Now you can, once you have this, you can convert you can visualize the actual aligned sequence from the multiple sequence alignment by converting it to a string, which you see here. You can see that there's a lot of gaps. All right. So this kind of brings us to the next function that we use, because oftentimes we want to refine the multiple sequence alignment from PFAM, because they can contain redundant sequences, or maybe want to filter out sequences that have just too many gaps. All right. So if you look up the documentation on refine MSA, you have things like the row occupancy and seek ID. All right. Row occupancy is basically going to filter out sequences with that have less than some provided threshold in terms of the number of gaps in that sequence of the aligned sequence. And then also with seek identity, seek ID keep unique sequences of a certain sequence identity level in your refined multiple sequence element. All right. So here, to create a new, I'm going to create a new refined multiple sequence alignment using refined MSA. And I'm going to provide a label, and then I'm setting the row occupancy to 0.8 and the sequence ID to 0.89. And this is going to filter our multiple sequence alignment based off of these specifications. And I'm going to slice this refined MSA, just pull out the first sequence. And we can see that the first sequence is now different than the original, because the original had its first sequence had a lot of gaps. Okay. All right. So now we're going to talk about actually trying to estimate conservation and coevolution. All right. And one really straightforward way of doing this is using the Shannon entropy, sometimes called the Shannon information content. And basically, it is a way of measuring the degree of uncertainty in the system that you are studying. And with respect to multiple sequence alignments, if you calculate the sequence entropy of a given position in the multiple sequence alignment, this is a way of trying to estimate how conserved a given position is. Okay, so if it is highly conserved, that means there are very few amino acid substitutions there, the entropy will be low. And if there's a lot of variability in a given position, that will give you a higher entropy, and the highest level of entropy you can get is if basically each amino acid in a given position occurs with a frequency of 1/20. All right, so it's basically like a uniform distribution, and that's basically telling you that at that site the protein can tolerate various substitutions there, and so it's not conserved. Yeah.
Could you turn off your camera? There's a synchronization problem. Either one of us has an internet connection problem or you have. Okay, please, thank you.
Yeah, sorry about that, guys. Yeah, so I've turned off my camera. Can you hear me better now?
Yes, thank you.
All right, yeah, thank you for letting me know, Burak. All right, so so just to stay on time, we're going to use the Shannon entropy to try to estimate and identify what regions of positions in the multiple sequence alignment are conserved. Okay, and this is conveniently computed using this function calc Shannon entropy that I'm showing here, and we're going to use the refined multiple sequence alignment. All right, and we can plot the Shannon entropy in this bar plot using show Shannon entropy. All right, and it's probably easier to see on this other screen here, but you'll basically get a plot like this. Okay. And moving forward, we can do we can go even further in trying to compare or tease out any statistical structure that may be present in our data of sequence evolution and structural dynamics. All right, and we're going to do this by first obtaining residue fluctuations for a single protein member of the above family using the Gaussian network model. All right, and so I'm going to be using this PDB file or this PDB ID, and we're going to we have to select for the chain B in this file. And if you don't have it, you can use fetch PDB to download it. All right. Now, what you have to do in this case is that you need to make sure that you select the right residues in the PDB structure that match the amino acids in the multiple sequence alignment that we have. All right. So there's another convenient function that does this very easy for us. Is align sequence to MSA, and with this you only need to hand it the alpha carbon atoms only, and that way you can get the non-repeating residue numbers, right? And I'm also, once I do that, I get the overlapping residues between these two, between the PDB structure and the aligned sequence from the MSA, and I'm using show alignment to visualize that here. Okay, and we can also take a look at the our residue selection. All right, and it's in this case you see we're showing the sequence from the PDB structure and the sequence from our protein sequence from the multiple sequence alignment, and it's clear here that we need to basically exclude these first two residues from the PDB structure. In Prody, all right, so now I've reduced my the object of my PDB structure to only the residues in the positions 3 to 121. Now, when we compare the two sequences from the multiple sequence alignments and our structure, they're the same. Now we can do the GNM analysis, which you all are familiar with now. I will plot this. All right, and I'm going to switch over here again. So in this plot, I think there might be a slight shift here between the plotted fluctuations and the Shannon entropy here. But typically what we find is that in many cases, the residues that have or that exhibit small fluctuations, it's not uncommon for them to be conserved residues, right? Because these proteins or these amino acids in the protein can help facilitate the large collective motions in the protein dynamics. All right. So moving on to coevolution now. This is also easily handled in Evol, and we have several functions here. The first one we'll take a look at is calculating the mutual information between the different sites or positions in the multiple sequence alignment. All right, and this is, again here, this is one way to estimate coevolution between two positions in the protein. And so I'm going to use the build mute info matrix, and I'm going to give it our refined multiple sequence alignment. And again, we have functions that allow us to visualize this data. All right. Now, this is kind of hard to interpret, and I'm going to show you several ways of improving the interpretability and the visualization here. So one thing we can do is to use the average product correlation, all right? Which again, there's a function that handles this for you. All right, and now we know it's easier to tease out some structure here. There looks like there's local dependency between nearby sites, and of course we can try to make this better by normalizing our color scale.
Is it my internet or your internet is a little?
Yes, we're seeing.
Okay, great. All right, yeah, I'll just finish up here. So there's another way to not only measure the dependence between residues, changing residues, but you can also use it to for contact prediction, which is this direct coupling analysis, and this is also provided it's also provided by functions in Evol, and it's again straightforward to use. So I will run this on the refined multiple sequence alignment, and again it's picking up some dependence between distant sites on the protein, and you can see these little hotspots in the off diagonal of this matrix. You can also try to validate whether or not it is a physical contact, or those two residues are in physical contact with each other by comparing it to the contact map. All right. And basically, to finish off, we can compute rank ordering statistics on the results from the direct coupling analysis, which is being shown here with the calc rank order. All right, so we're going to try to identify statistically significant residue pairs, which is what's being shown here. And we're going to do this for the direct coupling analysis and the estimates from mutual information. All right, so this was for the direct coupling analysis. This will be for mutual information. And you can see some agreement here between these residue pairs here, okay? And that's basically all I have for you today.
Okay. Hi, everybody. We are together again. So I will say a couple of things before starting. So this is the ESSA tutorial. If you have looked at the installation instructions, as you see, one part, let's say the second part of the ESSA tutorial, which is basically allosteric pocket prediction part, needs fpockets. And fpockets, if you install the fpocket, if you're using Linux or Mac machine, it's easy. But for the Windows machine, unfortunately, you cannot install fpocket easily, since all the CMake files, unfortunately, for Unix-based systems. So that's why we provided a workaround. If we could manage to do this workaround by installing a sub-Linux system and all the other required packages, you can start it from that instruction sets. But otherwise, if you couldn't do it, you can use your normal installed environment, and you can do everything till the pocket prediction part, and then you can just like watch the rest. Okay. So as you know, we need to first download the required Jupyter Notebook file, which is here. Either you can, as you know, download it here.
Burak,
Sorry, I cannot hear you, camera.
you're not sharing your screen.
Oh, I'm not sharing my screen. Oh, so sorry. Okay. Now you see it, right?
Yes, yes.
Okay. And let me make it bigger. Is it okay? Everybody can see it now?
Yes.
Okay, good. So either you can download the notebook file from here, or you can, as you know, you can go to Prodi website and then click the link here, and then you will end up with this workshops website, and you can download it here. Okay, you can just like save link as, and you can download it. Okay, and you, as you know, you have to start the Jupyter Notebook from that working directory. So I will do the same thing. Actually, I did it, so it's there. So is there anybody who couldn't download it or who couldn't start the Jupyter Notebook file for this tutorial? If this is the case, one of the project team member can help you, okay? Otherwise, I will continue. So, you know, till this day or this time, we always say that first we have to import all the required files, right? And I'm gonna do the same thing again. And now let me run this cell. As you remember, whenever you're in the cell becomes green. And then we basically now importing all the required packages. There's a little information here. The pandas is for data manipulation. If you are not doing any data manipulation on the data frames today, you don't need to import it. But otherwise, if you would like to change something, then you need to import pandas as well. So what we're going to do first, of course, like in the Prodi Basics or A&M tutorial or GNM tutorial or the other ones today, we first need to parse our structure, right? And the structure for today, for this session, is a beta lactamase, and its PDB ID is 1PZO, okay? And then now, since I already downloaded it, Prodi again tells me it found the PDB file in my working directory. So let's first, before going further, let me run VMD. Since this session is basically back and forth between VMD, let me open this structure in VMD. Okay?
Burak,
Yeah.
sorry. People asked you to, if possible, increase the size or font.
Sure, sure.
Thank you very much.
Okay. Is it better?
Yeah.
Okay.
Yeah, it's better.
Okay. Okay, now it's there. So what I'm going to do, I will go to my working directory and import or load this molecule. Okay. So what you're going to do, you have to first go VMD menu and then click File, and then after click this file, just click New Molecule, and this, as you remember, this windows pop up, and then you have to basically browse where the file is, and then go there. And I'm doing it. So, and then you have to choose that file. Okay. And then the only thing you have to do is just load it. And then now I have it. So I'm going to do the same customizations. I would like to remind you what you can do. You can change the color. And then I did it. Click graphics and colors. And then from display, choose background. And then choose white, for example. And then what you can do, you can just like leave it here, but you can change the basically display from perspective to orthographic. And then I will change the representation of this protein by click the graphics and representation. And then here you see there are coloring methods and drawing methods. I'm going to first choose new cartoon. Okay, and then now you see that's the cartoon representation of our molecule, right? I don't need this axis now, so I'm going to turn off the axis. So this, whenever you say draw me, if you choose the drawing method as new cartoon, you can just only see, the protein itself. But in this tutorial, this protein actually has two allosteric ligands, and their chain IDs are, I'm sure it's written here, 300 and 301. Okay, so how we can obtain or show those ones, you have to change your selection. Okay, now I know this is my protein, but I would like to see those ligands. So go to representation. This time, what we're going to do, we will click selection. And then it says all, but we would like to choose just the ligands. How I can do it? I can say residue ID 300 or residue ID 301. And I apply it, everything disappears. Since you cannot show the ligands in cartoon form, you have to change it. Let's choose dynamic bonds. Okay, so now I have those two ligands. Okay, let's leave it there. Okay, they can stay there, but I'm gonna also deactivate the display. Now I don't have anything. Okay, so let's continue. As you remember, everything in Prodi is an object. So in order to do an ESSA analysis, what I have to do, I have to initialize an ESSA object, and I'm going to do it. So now I basically initialized it, but again, this object is empty. So I have to put something in it, okay, to do the calculations. What I'm going to do, the first step is setting the system. Okay, what do I mean by setting the system? I have to first give the atom group. As you remember, when you parse a structure, the variable becomes your atom group. Okay, so what I have to do, I have to first give this atom group. Here, the variable name is PDB, it's okay. But as you remember, if you are using this function first time, or if you just used before, but you don't remember the parameters, what we're gonna do, we are basically doing the following thing. We are coming here, and inside the cell, Shift, Tab, Tab. It opens up not only the docstring, but also all the parameters it shows us. And let's look at what we need to, what we can set here. So the first one is the required variable or parameter, which is atoms, and we set it. And it says lig, and what it says, strings of ligands, chain IDs, and resonance separated by a white spaces. As you remember during the morning session, if you would like to show the ligand interacting residues of your protein, I said there is a way to create these profiles by also pointing out those residues. And also, as you remember, in ESSA, we are not actually doing anything with ligands, but we are mimicking the ligand, right? By means of this incorporation of the extra heavy atoms. But if you would like to show those residues, like ligand interacting residues, it's so easy. You can just give the chain ID and the residue numbers of those residues. If you provide this one, then you generate the ESSA profile, those ligand interacting residues will be shown on the profile. So that's why I'm also providing this thing, these ligand residue IDs and chains. And let's look at it. And if you remember my slide on the details of the ESSA, how we determine which residues are interacting with the ligands residues. It's easy. We need to give a spatial cutoff, and the default value is 4.5 angstrom. Okay, so I'm not going to change this thing. There is one more option. It's called lawman. The ESSA object is using a mode ensemble object in Prodi, but if you're, let's say, you are running an ESSA analysis on a large molecule, like for example spike, or your computer does not have enough resources, then you can also choose lawman option, and it will not generate a mode ensemble, but it will save all the eigenvalues and eigenvectors. Okay, for this one it's okay, there is no problem, so we don't need to give this flag true. So what I'm going to do, I'm just setting the system. Now I set the system. Prodi knows there's an ESSA object, the atom group of the proteins is set, and also we provide the ligand IDs to show, okay, on the profile. The next step is so easy. The function to scan those residues is scan residues, and there are a couple of parameters. You see, it says the number of modes to evaluate the mean shifts is 10. If you would like to, let's say, do this ESSA analysis based on the first non-zero 3 modes, then you can set it 3. Or if you would like to use the first 20 modes, you can set 20. And the default elastic network model for ESSA analysis is GNM. If you would like to use an isotropic network model or ANM, then you can just write here ANM. And if you don't specify the cutoff for the specific elastic network model, as you remember from yesterday, the cutoff value for GNM is 10 angstrom and 15 angstrom for ANM. So if you don't specify it, it will just fetch those values and use those default values, okay? And it's so easy peasy. What you're gonna do, you just like, since I am in the cell, I can hit Shift Enter, and then it starts scanning. Of course, it depends on your computer, okay? So till this end, everybody is with me and everybody can see the scanning on his or her screen. And I think there is couple of things in the chat. Let me go. Okay, I saw it. So nice. So as you see, it's very efficient. It just creates those perturbed and unperturbed models, and then it will evaluate everything you need. So now I have basically all I need. So I can see the profile. The easy way to see the profile is the function called show ESSA profile. Here I'm doing a bit intermediate matplotlib customization. I will change the DPI of the figure with a context manager. Okay, you don't need to do this thing, but it's good to know. Okay, and then what I'm going to do, I'm just like run this command, and then it will generate this figure, as you see. So now, if you remember, this blue line represents the boundary between the top 25 percentile of the z-scores and the rest. And since we provide the labels, chain ID and the residue number of the ligands, it also shows the protein residues which interact with those ligand residues, right? It's very nice. And as you see, the plot automatically uses the function show atomic lines. We saw that function yesterday. It puts the correct residue numbers which are written in the PDB file. But we can even further customize this plot. How we can do it? For example, you can change the figure size. This line still matplotlib customization. But the important thing, let me put this thing here. What are the parameters of this show ESSA profile function? Let's look at it. I'm just scrolling at it, and then shift tab. Now I can see. So it says the quantile value. So you can change this quantile value. For example, you can say, oh, okay, I'm just interested in the of the residues in the first 5 quantile, right? Top 5 quantile. You can change it. Or what you would like to see, you can see here, for example, in this figure, you can just see the circles representing those residues, but you would like to know the single letter code and the residue number. Then the only thing you have to do is using this flag. If you say res code is equal to true, then it will show up those ones as well. And let's say from your experience, you know some of the residues are important and you would like to put those residues on the profile as well to see that they can change or alter the dispersion of the vibration spectrum or not. It's also easy. The only thing you need to do is using ProD selection algebra and set those residues you would like to see to the parameter called cell, stands for a selection. So I'm going to do this thing. And then when I did it, as you see, this res code says the show ESSA profile method of function, please include the residue codes. And since I'm using here almost the 5 top percentile, it just says, okay, don't deliver or show the residue codes of the residues which are below this boundary. But if you would like to see the specific residue which you basically give in the selection algebra, it overrides and still it puts there. So this, in this case, it is that residue. Basically, as you see, it's so easy that this module, it evaluated the scores for you, and also it gives an intuitive figure. It generates an intuitive figure for you. Of course, for publication or production purposes, you can regenerate this figure as you like, okay? But of course, you don't need to run the ESSA scanning algorithm all the time. So you would like to use the results. What you would like to see, what you would like to do then, you can just save or store those ESSA scores. And the function for it is ESSA save ESSA z-scores. It will basically save all the z-scores as a NumPy array for you. And whenever you did this thing, let me go to the working directory, you see it basically generates this file. It's a NumPy file. So after, let's say, a couple of days, you can just load this file in terms of NumPy load file. Let's say you show the profile, see the profile, but you would like to see the values of this ESSA scores. How we can do it? You can use this getter function. It says, give me the ESSA z-scores, okay? Whenever you see a get in front of something after the dot, it means that it's a getter function. okay? And you can also slice it since it's a NumPy array. I'm just slicing and getting the first 10 entries of this NumPy array. But of course, we would like to color code this protein in terms of these SLZ scores, right? So since you also saw those nice figures in the morning session, how you can do it? You can just use write SLZ scores to PDB. It will extend those values, since these are based on carbon alphas or residues, to the whole atoms, and then it will create a PDB file with the name of this or title of this PDB. And it will also add something to that, some identifier to the PDB file. So let's do it. Okay, let me go back to working directory. As you see, it created this file. So this is the title of the PDB or the name of the PDB, and it puts GNM since we use GNM, and it says this is the Z-score, and then it basically creates this PDB file. Let's load this PDB file in VM. What I'm going to do, I will come here, again, new molecule, and then browse, and then choose this PDB file, and open up, load it. Now it is there, okay, as represented as lines. So I would like to make it first as cartoon. Okay, I'll change the drawing methods. So this is my protein, as you see. But this time, what I did, as you remember, I saved all the Z-scores to the B factor column, so I can choose the coloring methods as beta, and then now I have it. So, as you remember from yesterday, I would like to change the color scale, okay? And then I'm going to rainbow, and then use this one. You can choose whatever you would like to, okay? You can choose, for example, from blue to reds, so which means that the red ones are the residues with high Z-scores, okay, and the blue ones is the low Z-scores, but I'm going to choose rainbow. If you don't have rainbow, it's okay, since this scale was added recently, I think. So if you don't have it, no problem. So now I have the structure, right? I color coded with the SLZ scores. It looks nice, but let me activate the original PDB. Since original PDB has the legend information, but those created files by ESSA only has the protein residues. So I will activate it, and you see it's somewhere there. It's somewhere there, okay? And in order to understand better, even I can change this representation from new cartoon to surface. Wow, as you see, so that region is hot, it's orange. If I looked at the color scale. So, but I would like to further customize this one, how I can do it. You can change the material. Do you remember from yesterday? I can choose chalky, which is better, okay? And then, it's very nice. They're somewhere here. So as you see, there is one red region here. If I go back to cartoon representation, so it's basically an end of a helix. That's why this is the case. So, and it's also a bit like overshadow our results. Okay. So this is the first part of ESSA. So the second part of ESSA is the prediction part of it, right? In order to do that, as I said, you have to have the F pockets. If you don't have the F pockets now, just watch it. If you have the F pockets, it's so easy to scan the residues. The only thing you have to do is write this command and run it. And it will basically fetch some of the files which ESSA generated and then apply F pockets at the backend, okay? And then it creates this directory. This directory contains lots of things about the pockets. Okay, so there are some scripts which we will use, and also some PDB files which tells the features, what kind of a feature F pockets obtained for those pockets, or what are the coordinates of the alpha spheres. It uses alpha spheres. Also, it uses some residue informations. Details are not important for this session. Now I know it created this directory and all the PDB files which I need. What I can do, would like to see those z-scores, right? Since what ESSA does, it will assign those ESSA z-scores to the pockets as well in order to rank them. So let's look at the pockets. Since this is a highly small molecule, the F pocket found just seven pockets here, okay? This is actually a pandas data frame. If you don't do any manipulation on it, as I said, you don't need to import pandas, since scan pockets automatically import panda. But whenever you would like to do something more, then of course you have to also explicitly import panda in your Jupyter Notebook file or in your session. So we provide three scores here. All of them are z-scores. As you see, we have lots of z-scores for each residues participating in the pockets. Sometimes some part of the pocket can be around a hinge residue, sometimes not. So that's why we are providing two z-scores, one of which is the maximum z-score for that specific pocket, okay, or the median z-score. And also, for example, here we have 7 pockets. We are fetching all the informations from pockets, and then like the LHT scores, and we are evaluating the z-score for LHTs in terms of those pockets, okay, overall pockets. So now we have those values. Even if you don't rank them automatically, you can see. And what was the criterion for us to exclude any pockets? If the z-score, LHT z-score is less than 0, we basically exclude it, right? It's a hard cutoff, and as I said, it's not a good one, but it works fine. So what I'm going to do, I will rank them. So the ranking is based on, it will exclude the pockets with negative LHT scores and then use a hierarchical ranking. It's called leg sort. And I will do it. It ranked. And then whenever I ask, give me the pocket rank, since there is just one pocket with positive LHT z-score, it just says, okay, this is the pocket with high potential to be an allosteric site or allosteric pockets, okay? So now I'm gonna, of course, this is not the case. For example, if you're working on a large molecule, you can end up with 100 pockets, and then after ranking, you end up with maybe 20 or 40 pockets. Then you have to take the first 5 or first 10 of them, depending on the size of your protein. So we basically get all the pocket ranks. What I can do, I would like to see where those pockets, right? It's easier in PyMol, but we are not using PyMol in this workshop. So we will do this thing with VMD. It will be a bit, let's say, intermediate, but you can do it. So what I have to do, I have to run one of those files, either the vmd.sh, pymol.sh, or I can use this PQR file, or I can use TCL. And as you know, VMD uses TCL. And that's why I'm gonna run this one, okay? So what I have to do, I'll need to come here and then choose TK console, okay? Is everybody with me till now? I don't see anything in the chat. I suppose you're following it. So let me check, where am I? I'm just here. I will go to desktop since my file is in desktop, and you have to go to that place wherever your Jupyter file and these underscore out files are, okay? and then workshop, and ESSA. The name is 1PZO and Pro, and out. Now I'm there. Since I'm there, I can run this TCR file. What we will do, since I'm in the TK console, I'll go File, and then choose Load File. I will go here, and then choose this TCL file. And then it comes like this. I have a problem with rendering on this machine. That's why it looks like this. There are 3 options for rendering, okay? And you can change that one. For example, this TCR command basically says use GLSL, and it does not work on my machine. So I will choose Acrobat 3D. This one is heavier, but it's okay. Now I have overlaid proteins and dots and lots of things. Let me do the following. First, what I'm going to do, it automatically, as you see, loads the PDB file. I'm not going to use this one, so I'm going to deactivate it. Okay. You see there are pockets there. How many pockets I have? If you remember, I have 7 pockets. Let me go. Yeah, I have total 7 pockets. And the pocket 6, the 6th one is the one with high allosteric potential. So then I would like to see that one. What I'm going to do, first I'm going to customize this one a bit more. Go to display settings. And then I will change Q mode to linear to get rid of shadowing. Now I don't have any shadowing. Now, as you see, it's really nice. And then I will choose first representation. It's there. I don't want to have those points. As you see, there were a couple of points here. I'm not going to go into details what they are, but I'll just like deactivate them. And the quick surf is basically those pockets. Okay. So what I'm going to do, since I know the 6th one is the top ranking pockets, I will say res ID 6. And it's now here. And in order to see it better, I will also change its color. How I can do it? You can come here. You can change this coloring method to color ID. And then this one changed. You can come and I will choose magenta since I like magenta. Okay, now it's there. But in order to recognize its location on the protein, it's better to change my results color coded with Z-score from cartoon to surface representation. It's very easy to do that. In the selected molecule, you can choose your result and make it surface. And then, as you see, the 6th pocket is there. But at the beginning of this session, I also load the original PDB file, which contains the ligands. Then what I'm going to do, as you remember, I changed the selection for that specific original PDB to just see the ligands. I'm going to activate it. Bam, it's there. As you see, we did a very fast and efficient scanning. And then we used a couple of information more, like the one is LHD Z-score. And then we exclude some of the pockets. In this case, almost all of them except one. And the pocket we end up with is the one which has the most allosteric potential, and it accommodates those allosteric ligands. Whenever you see such a thing, which means that you hit the jackpot, since you found the correct pockets. And let's say you did it and you're totally happy about your results, what you can do, you can save those results as a pickle file. In order to do that, you type this command, save pocket z-scores, run it, and it will basically save all your results here in the data frame. You cannot, either you have to write your own code, or you can use the pickle library in Python. Since it's a data frame, it has lots of information. It's better to save it or store it as a pickle file. But at the same time, you don't want, since it's a binary file, you would like to have the result as a CSV. That's why we also implemented this function. It basically stores your ranking as a CSV file. And if you go back to your working directory, now it is there. And if you click it, you will see it. And that's all about the